Yeah, please proceed, ma'am.
So, hello everyone, welcome to Tawhid Foundation at Allied Healthcare Sciences, Bangalore. Today's class is about parasitology, an essential branch of science in the study of health and diseases. So, parasitology is the scientific study of parasites, their host, and the complex relationship that exists between them. Parasites are organisms such as protozoa and worms that depends on other living beings, often humans for survival, sometimes causing diseases like malaria, filariasis, or intestinal infections. So, understanding parasitology equips one to identify important parasites, learn about their life cycles, diagnose related diseases, and help prevent and control their spread through laboratory skills and public health measures. So, for today's class, parasitology for BSc MLT first year students. Scope and importance. This slide introduces the overall goals of parasitology. It emphasizes the need to understand parasitic infection, including their diagnosis and ways to prevent and control them. It also highlights learning the life cycles of important parasites, preparing lab reagents, and applying diagnostic procedures in a laboratory settings. Objectives. So the objectives focuses on understanding terminology in parasitology, learning about different parasites and the diseases they cause, and gaining knowledge about laboratory diagnosis methods. It includes awareness of basic epidemiological aspects, techniques for specimen handling and processing, and last, the importance of infection and prevention and control measures. So, definition of parasitology. Definitions are provided for key terms such as parasitology, the scientific study of parasites, medical parasitology, the study of human infecting parasites, and the term parasite itself, which refers to any organism dependent on a host for survival and causing potential harm. So, Francisco Reddy is acknowledged as the father of parasitology. Host and carrier. So this slide explains the concept of host, which are organisms that supports parasites. It distinguishes definitive hosts where parasites mature and reproduce sexually from intermediate hosts where development occurs but not sexual production. It also briefly defines carriers, which are organisms that transfer disease causing agents to new hosts. Here you can give example of parasite organism dependent on host, that is Plasmodium vivax. Most common example. Relationships. The various biological relationships are explained here. First is symbiosis, involves two organisms living together, sometimes benefiting one or both without harm. Commensalism. Commensalism benefits one partner without affecting the other. Mutualism. Mutualism benefits both, and parasitism benefits the parasites while damaging the host. So, major parasites and diseases. An overview table lists common parasitic organisms and the diseases they cause. Example include malaria from Plasmodium vivax, pneumonitis, intestinal obstruction from roundworm, that is Ascaris. Blood diarrhea or rectal prolapse is caused from whipworm, that is Trichuris. Coughing, abdominal pain, anemia is caused by hookworm and Xylostoma necator. Renal intestinal diseases, cystosoma caused by parasite. Lymphatic filariasis, elephantiasis is caused by filariae. Chagas diseases, cardiovascular is caused by Trypanosoma cruzi. Cutaneous, mucocutaneous and visceral leishmaniasis caused by Leishmania parasites. So these are the examples of parasites, types of parasites. This disease is caused by parasite called as filariasis. Here you can see the swollen legs. It is caused during rain. Mucocutaneous leishmaniasis. This infection is caused by parasites. Giardia intestinalis. So here this figure most likely depicts the microscopic appearance of Giardia intestinalis, a flagellated protozoan parasite. Giardia is known for its characteristic teardrop or pear-shaped structure and has flagella that allow it to move. So in clinical diagnosis, seeing the trophozoite or cyst form in fecal samples under the microscopy is key for confirming giardiasis, which causes gastrointestinal symptoms such as diarrhea and abdominal cramps. So these days, diarrhea is very common, caused by Giardia intestinalis. Plasmodium falciparum in erythrocytes, which is red blood cells. So these diagram display red blood cell infected by the malaria parasite. Plasmodium falciparum, it showed different parasite stages such as ringworm, trophozoite, schizont, and gametocyte. Inside the erythrocyte, identifying these stages in a blood smear is crucial for diagnosing malaria and differentiating Plasmodium species. Since Plasmodium falciparum is responsible for the most severe and life-threatening type of malaria. Classification of parasites. First is protozoa. Protozoa are described as unicellular eukaryotic microorganism lacking photosynthetic ability. They use flagella or cilia for movement and thrive in moist environments, making them an important group in the study of infectious diseases. So second is helminthes. Helminthes are detailed as parasitic worms that usually live in the intestines. They have bilateral symmetry, reduced digestive and nervous systems, and produce a large number of eggs, reflecting adaptations for survival and propagation inside host. Protozoa. So protozoa are unicellular eukaryotes with no photosynthetic ability. These are heterotrophic nutrition, indefinite shape, size, motility. They do movement by with using flagella or cilia, live in moist habitat, and it is studied in protozoology. Pathogenic protozoan, parasites of men. So this diagram illustrates several pathogenic protozoa that infect humans such as Giardia lambelia. Giardia, first I explained Giardia intestinalis. It causes diarrhea. Leishmania species, these Leishmania species, Entamoeba histolytica, this one Entamoeba histolytica, Plasmodium vivax, Cryptosporidium pavium, and Trichomonas vaginalis. Each protozoan would be shown in this typical shape. So example, Giardia appear as a pear-shaped organism with flagella, Plasmodium with red blood cell, and Entamoeba with its amoeboid appearance. So visual identification is a core skill in laboratory practice for the diagnosis and differentiation of protozoan diseases. Protozoan diseases. So a table to link protozoan parasites with corresponding diseases such as Giardia causing giardiasis, Plasmodium causing malaria, Leishmania tropica, cutaneous leishmaniasis, Leishmania donovani caused diseases, Kala Azar, which is visceral leish, Entamoeba histolytica, it causes diseases that is amoebic dysentery, Cryptosporidium parvum, it causes cryptosporidiasis. Last is Trichomonas vaginalis, it causes diseases vaginal trichomoniasis. Helminthes, its general features. So helminthes are detailed as parasitic worms that usually live in the intestine. They have bilateral symmetry, reduced digestive and nervous system, and produce large number of eggs, reflecting adaptations for survival and propagation inside the host. Platyhelminthes and nematodes. So important worm species and their associated diseases are presented, such as schistosoma causing schistosomiasis, that is liver, lung, gut and bladder diseases. Echinococcus causes hydatid diseases. Taenia solium, Taenia saginata, Diphyllobothrium latum. This causes diseases like tapeworm infections, which caused in pig, cow, or fish. Nectar trichuris, it causes diseases from hookworm, whipworm, that is gut infection. Last is Wucheria onchocerca. It causes elephantiasis, river blindness, and filariasis. Classification of human parasites. So human parasites are further classified into helminthes, that is nematodes, that is roundworms, causes intestinal or blood tissue infection. Cestodes, that is tapeworms, caused intestinal and tissue larval stage infection. Trematodes, that is flukes, caused intestinal, liver, lung, or blood infections. Protozoa, intestinal, that is, it includes amoeba, flagellates, ciliates, epicomplexa. Blood and tissue, that is flagellates, amoeba, epicomplexa. So outlining the broad spectrum that affects humans. Nomenclature of parasites, the use of binomial nomenclature for naming parasite. It includes examples of protozoa and helminthes names. So showing how scientific classification aids in clarity and consistency when identifying and discussing parasites. Protozoa, Saccharomyces includes Entamoeba histolytica, Giardia lamblia. Epicomplexa include Plasmodium vivax, Plasmodium malaria, Plasmodium falciparum. Coleophora include Balantidium coli. Microspora causes enterocytosome by Enuzi E. element encephalitozoon cuniculi. Platyhelminthes cestoda, this causes Diphyllobothrium latum, Taenia solium. Trematodes, this Mansoni and S. japonicum. Nemathelminthes ascaris, lumbricoides. Parasitic infection. So it defines a parasitic infection as any disease caused or transmitted by a parasite. Understanding this concept is crucial for recognizing their impact on health. Sources of infection. Possible sources of parasitic infection include contaminated water, ingesting eggs in food, arthropod bites, and direct penetration through skin or mucous membrane. So these highlights the various routes by which people can get infected for parasites. Modes of transmission. So modes of transmission is summarized in a table showing that parasite may be spread via oral infection, oral induction, that is vector like insects, sexual contact, inhalation, or direct contact, skin or types. Vector borne, it causes again these diseases. Inhalation, it causes these diseases. Skin and eye contact causes these diseases. General classification of parasites. So parasites are classified based on their interaction with host. Ectoparasites live outside the body, endoparasite inside. Some are temporary or permanent, others are facultative or obligatory. Some wandering parasites and occasional parasites. So it affects ecto affects outside, endo affects inside the body. Temporary, it will visit for short period of time. Permanent, stays for entire lifespan. Facultative, only when favorable opportunity arises. Obligatory needs living host always wandering a typical locations, occasional affects unusual host. So overview of the life cycle. So here mosquito stage, human liver stage, human blood stage, mosquito uptake stage, and last is mosquito gut to salivary gland stage. So in mosquito stage, the cycle begins when an infected female Anopheles mosquito takes blood meal from a human and inject sporozoites, that is infective form, into bloodstreams. So the sporozoid travel to the human liver and invade liver cells, starting the exoerythrocyte cycles. So in the liver, sporocytes develop into schizons, which eventually rupture, releasing thousands of merozoites into the bloodstream. So human liver stage. Upon entering the liver cells, sporozoites mature into schizons. The schizons rupture, releasing merozoites that invade red blood cell, beginning the erythrocytic cycles. So, human blood stage, inside red blood cell, the merozoite develop into immature trophozoites, that is ring structure. These trophozoites mature and form schizons again, which rupture and release more merozoites, continuing the infection cycle within red blood cells. So, some merozoites differentiate into gametocytes, that is male and female, these gametocytes. So, mosquito uptake stage, when mosquito bites an infected human, it digests these gametocytes in the mosquito's gut, that is male microgametes and female macrogametes. Gametocyte fertilize, forming a zygote that transforms into motile ookinates. So ookinates penetrate the gut lining and develop into oocyst. Oocysts grow, rupture and release thousands of sporozoites. Again, this releases. So sporozoites migrate to the mosquito salivary gland, ready to infect the next human when the mosquito bites again, which completes the cycle. So the parasites alternates between human and mosquitoes. In humans, the parasites multiply mainly in the liver part and red blood cells. In the mosquito, reproduction occurs producing new sporozoites. Here, sporozoitic cycles. Understanding this cycle is essential for malaria diagnosis, treatment, and control strategies. So the life cycle explains the persistence and spread of malaria and guides biomedical approaches to interrupt the parasitics development and transmission. So these diagrams represent a classification of laboratory tests using in parasitology and clinical diagnosis organized by sample type, which includes stool, urine, or blood. Classification. So these diagrams represent a classification of laboratory tests used in parasitology and clinical diagnosis organized by sample type, which includes stool, urine, and blood. So stool samples, stool analysis is divided into three categories: physical, chemical, and microscopic. So in physical test, color and odor, consistency, checks whether the stool is formed watery or hard, accesses for abnormal changes that indicate diseases. So mucus presence may indicate infection or inflammation. Chemical test, pH determines acidity or alkalinity altered in various conditions. Reducing substances, check for sugars like glucose or fructose indicating malabsorption. Microscopic test, leukocytes detects white blood cells, markers or infection of inflammation. RBCs, red blood cell presence may indicate bleeding, ova and parasites. So in identification of parasite eggs or organisms, meat fibers, muscle fibers indicate digestion issues. Fat presence may suggest malabsorption or pancreatic diseases. For urine analysis is categorized again into physical, chemical, and microscopic. So physical test, color and odor changes may signal infection or metabolic problem. Turbidity, cloudiness suggests presence of cells or crystals. Specific gravity measures urine concentration. pH, urine acidity, alkalinity altered in infection or diet. Chemical test, protein usually absent, presence suggests kidney issues. Glucose, so indicate diabetes or kidney problems. Ketones, nitrates, markers of metabolic stages and bacterial infection. Urobilinogen, bilirubin, liver function indicators. Blood present indicate bleeding or damage. In microscopic, RBCs and WBCs detect infection and inflammation. Epithelial cells accesses contamination or pathology. Microorganism presence of bacteria, yeast or parasites. Last is blood test. Thin and thick smear used primarily for detecting blood parasites. Example, malaria. Thin smears helps identify species. Thick smear detects presence and density of parasites. So laboratory sample collection. So guidelines for collecting stool samples are presented, including sufficient quantity, proper container, immediate examination, and avoiding urine contamination. These steps ensure accurate lab results for parasitic diagnosis. Instruments used in labs. So these are the instrument which is used in lab, sterile containers, labels, etc. to avoid confusion between samples. Physical examination of stools. So here different aspects of stools such as color, odor, quantity and consistency are explained as indicators of health or diseases. Abnormal findings, example black color, foul odor, mucus presence, can point to specific conditions like dysentery or malabsorption. Stool examination method. So two main smear techniques are described. Saline smear reveals egg and motile protozoa, while iodine smears are better for seeing cyst morphology. These are basic laboratory methods for parasite infection. So here in saline smear, huge number of eggs, that is protozoal trough, motility, amoeba flagellate presence. In iodine smear, huge number of cyst morphological details will be seen. Chemical examination. So stool chemistry can help spot understanding issues. Normal stool pH is close to neutral. Deviations may signal malabsorption, infection, or other condition. So the presence of reducing sugar may show poor carbohydrate absorption or viral illness after gastroenteritis. Microscopic examination. So this detailed list of findings in stool microscopy includes leukocytes suggesting infections, red blood cells indicating bleeding, ova and parasites confirming infection, undigested meat, muscle fibers, malabsorption, and fat digestive enzyme or bile deficiency. These findings are crucial for diagnosis. Serodiagnosis. So serodiagnosis used blood tests like IHA and ELISA to detect antibodies or antigens against parasites, especially useful in invasive or non-obvious cases, as these tests offer a higher sensitivity than basic microscopy. Stool culture. The process of culturing stool samples to identify parasites is described, noting that specific media are used for growing organism in suspected chronic or hidden infection, improving detection accuracy. Fecal occult blood test. So these tests detect even tiny amounts of blood in stool that are invisible to the naked eyes, which may signal intestinal bleeding due to parasitic or pathological processes. Urine analysis. Urine Analysis, physical examination. So starts with basic checks for volume, color, and odor. Changes can indicate disease such as increased volume in polyuria or a strong ammonia smell when bacteria are present, gives clues to underlying issues. Reactions and specific gravity. Urine pH and specific gravity are measured to assess kidney and metabolic health. Deviations from normal can signal diabetes, kidney diseases, excessive water loss, or dietary factors. Appearance and sediment formation. So normal urine is clear. Cloudiness or sediment points to infection. Phosphate crystals or other components. Milky urine may signal fat and sediment forms from cells or crystals setting at the containers of bottom. Urine analysis chemical test. So urine is checked for protein, glucose, urobilinogen, ketones, bilirubin, blood, nitrite, and leukocytes. So if protein is present, it indicates kidney diseases, stones, myeloma, anemia. Glucose is present, it indicates diabetes, kidney, hormonal, liver disorders. Ketones, fat metabolism, diabetic condition. Urobilinogen positive for hepatitis, cirrhosis compared to bilirubin. Bilirubin early indicator of hepatitis, liver and jaundice. Blood hemoglobin increases for UTI, kidney diseases, trauma or exercise. Nitrite indicates bacteria in UTI and leukocytes indicates kidney urinary tract inflammation or infection. Microscopic urine examination. So urine is centrifuged and analyzed for red and white blood cell, epithelial cell, microorganisms, and crystals. So these findings reveal infections, inflammation, and kidney disorders, helping pinpoint cause and type of illness. So these microscopic examination of urine. Normal crystal is uric acid, calcium oxalate, hippuric, calcium phosphate, triphosphate, calcium carbonate, ammonia blurate. So, if the person is infected, urine will contain all these crystals. Abnormal crystals are bilirubin, cholesterol, cysteine, leucine, tyrosine, sulfur, all these crystals will be present. A variety of normal and abnormal crystals may be present in the urine sediments in sterile containers. Microscopic examination of urine. You can see here red blood cells in urine appear as refractile disc, white blood cells in urine. These epithelial cells in urine. Diagrammatic representation of microscopic examination. Urine analysis by three methods: physical examination, microscopic test, biochemical examination. Physical examination includes volume, color, odor, pH, and specific gravity, whereas microscopic tests include cells, crystals, microorganisms, parasites, contamination. Biochemical examination include protein, sugars, ketone bodies, bile salts, bile pigments, and bloods. Blood test for parasite detection. So blood smears, thick and thin, are used to detect malaria and other blood parasites. The thin smear helps specific identification, while the thick smear detects parasites presence and counts parasitemia. Blood film stains. So various stains are used on blood films to highlight cell types and abnormalities. These stains such as Giemsa and Leishman's aid in distinguishing between different parasites and blood cell changes. Thick and thin smear preparation. So preparation methods for both thick and thin smear are described. So how samples are made, fixed and stained, ensuring optimal visualization of parasites and accurate diagnosis. Blood smear overview. This slide clarifies the specific uses. So thick blood smear reveals parasites present and thin smear confirms the species, essential in malaria diagnosis and treatment planning. Blood smear overview. Again, as I mentioned here, thick smear and thin smear. Prevention and control. Prevention strategies including educating the public about parasite risk, improving water quality and disposal of animal waste, and promoting hand hygiene to avoid infections from food, water, and animals. Additional prevention measures, practical tips are offered like nail hygiene and toilet use precaution, wearing shoes to avoid soil-borne parasites, and using sterilized solution for contact lenses to block parasite entry. Laboratory safety rules, basic lab safety is stressed. Avoid food and drink, wear protective gear, prevent direct contact with chemicals, avoid mouth pipetting, clean workspace, and quickly report and clean any chemical spills, making for a safety laboratory environment. Laboratory workflow. So there is three method we can follow for laboratory workflow: pre-analytic, analytic, and post-analytic. So patient preparation, sample collection must include proper care in sterile condition, sample receipt, sample transport in hygienic condition, quality control testing. Last is record keeping and reporting. Um, so that's all for today. For question and answer, that
Yeah.
slide is not 2 minutes, sir.
Okay.
1 second. Hello.
Hello.
Hello. Am I audible, sir?
Yeah, please.
My screen is visible, sir.
No.
1 second, sir.
When should we start then?
I don't know why it is not. I am connected, sir.
No, madam.
It is not connected.
So maybe
Yes, sir.
internet issue,
Yeah,
madam.
no, sir. I'll 1 second, sir. It is fixed.
Yeah.
So for 3 marks question, there are 5 questions. List the physical and chemical. Oh, my screen is not. List the physical, chemical, and microscopic tests performed on stool samples in parasitology. Second is name any 4 chemical tests done on urine samples and mention their significance. Third is what are the 2 types of blood smear used in parasitology, that is thick and thin smear. State their purpose briefly. Explain the importance of examining consistency and color in stool samples. Last is mention 2 microscopic elements examined in urine analysis and state their clinical importance. For 5 mark questions, describe in detail the various tests performed on stool samples for detecting parasitic infections and their diagnostic relevance. Second is explain the categories of urine examination tests and discuss how they help in diagnostic parasitic or other infections. Third, compare and contrast thick and thin smears used in malaria diagnosis, including their preparation and diagnostic value. Fourth, discuss the role of microscopic examination in stool and urine analysis in parasitology. Include example of each element searched for and what their presence indicates. Last is outline a schematic approach to laboratory analysis of stool, urine, and blood samples for parasitic infections, highlighting the types of tests and their significance. So that's all for today's class.
Yeah, wonderful presentation, madam.
Thank you, sir.
Parasitology. You have
Yeah.
covered, I think, basic parasitology. So
Yes, sir.
introduction to parasitology.
Yes, sir.
And coming
The
classes, I think we'll go individual parasites.
individual
Okay.
parasites. Their life
Yeah,
cycles and
individuals.
introduction I have covered.
Yeah, yeah,
Second class, I'll try to mention everything
yeah,
and
yeah.
related topic to that.
Thank you, madam. Thank you so much.
Thank you so
Yeah,
much,
yeah.
sir.
Done, done. Thank you, madam. Thank you so
Thank
much.
you.
At least